I'm Catherine Mishka, and this is the case study for Unit 4. So in the first scenario, our patient is positive for branched-chain amino acids in the blood. This is indicative of a disorder referred to as maple syrup urine disease, as the keto acids excreted via urine give it a maple syrup smell. In MSUD, it's a genetic disorder which results in a decrease or complete lack of activity of the branched-chain alpha keto acid dehydrogenase complex, or BCKAD, which is responsible for the breakdown of the amino acids leucine, isoleucine, and valine. BCKAD is an enzyme complex that's composed of 3 different catabolic subunits and a transaminase. Branched-chain aminotransferase, along with alpha-ketoglutarate, convert leucine, isoleucine, and valine into their alpha-keto acids. The E1 subunit will then decarboxylate the alpha-keto acids, and once it's decarboxylated, the E2 subunit, which contains a lipoic acid residue, will then transfer the acyl group contained in E1 to CoA, and then the E3 subunit will then reoxidize the lipoic acid. The alpha keto acids are metabolized into an intermediate form, and the activity of BCKAD phosphatase and BCKAD kinase will then further metabolize the alpha keto acid intermediaries into succinyl-CoA, acetoacetate, and acetyl-CoA, which we know are important for a number of metabolic processes. Any disruption in the enzymatic pathway or complex results in the incomplete degradation of these 3 essential amino acids and their intermediates and leads to a toxic buildup in the system. So in the brain, BCAA metabolism is responsible for glutamate maintenance, which is a critical neurotransmitter in the central nervous system and responsible for structural and cognitive development. And abnormal BCAA metabolism will cause neurologic complications. Inability to break down leucine and the resultant buildup affects water homeostasis in the brain, which causes edema. It also affects nitrogen homeostasis, which leads to a further decrease in glutamate levels. And also, excessive leucine will compete with other necessary amino acids in the central nervous system, such as tyrosine, which in turn affects protein signaling. There are 5 different types of MSUD: classic, intermediate, intermittent, thymine responsive, and E3 deficient. In classic, less than 2% of the BCKAD enzymatic complex is still functional. This form of the disease is typically diagnosed through routine screening in the neonatal period, but if undiagnosed via screening, symptoms most often occur within the first weeks of life and include the telltale presence of maple syrup scented urine and cerumen. Additionally, the neonate will begin to develop lethargy, poor feeding, irritability, and apnea, and if left untreated, coma and death related to the cerebral edema caused by leucine buildup. Acute neurologic decompensation may also occur and is typically a result of endogenous protein catabolism, which is present in high fever states, infections, exercise, or bodily trauma, including surgery. In intermediate MSUD, the BCKAD complex is still about 30% functional, and individuals are typically asymptomatic or may only have characteristic maple syrup scented urine, with additional neurologic and feeding issues resulting a little bit later on in life, usually during the first year. Intermittent MSUD sufferers are completely asymptomatic with normal neurologic functioning and growth, and symptoms only occur during increased catabolic states, like we said above. And symptoms are the same as in classic MSUD. So these 3 forms of MSUD are all a result in defects of the E1 and E2 subunits of the BCKAD complex. A 4th form is a result of a defect of the E2 subunit and is caused by a defect in the dihydrolipoamide branched-chain transacylase gene. This form of MSUD is referred to as thymine responsive and is the rarest of the 5 phenotypes. Symptoms are the same as in the other 3 forms, but have been shown to be responsive to thymine supplementation. So lastly, E3 deficient MSUD is the 5th and final variant of the disorder. It is considered dihydrolipoamide dehydrogenase deficiency, which is a subunit responsible for a bunch of different enzymatic processes. And because of this, E3's importance in all these metabolic complexes cause a host of other symptoms, including the neurologic symptoms. So these sufferers also experience liver dysfunction, eventual liver failure, encephalopathy, and eventual death. Nutritional therapy is critical in management of MSUD and focuses primarily on dietary modifications, as the affected amino acids are all essential amino acids that cannot be synthesized and have to be taken in via the diet. So diets focus on restricting branched-chain amino acid intake, It asks if there are any amino acids that become essential as a result of deficiencies present, and no, there are none. But as stated above, the affected amino acids in MSUD are already considered essential, and medical foods free of BCAAs can provide a majority of the protein needs while completely removing those BCAAs. And then valine and isoleucine supplements are added to maintain appropriate levels, and then leucine levels are monitored to ensure they don't reach toxic levels. Lastly, the question posed what vitamin supplements may be useful in the treatment of the suspected disorder. And it's just, again, thymine supplementation has shown to be beneficial, especially in the thymine responsive type. In the second scenario presented in this case study, this patient is presenting with symptoms of a deficiency in homogentisic acid oxidase and is referred to as alkaptonuria. Alkaptonuria is a genetic disorder that results in the disruption of tyrosine degradation. Tyrosine is a non-essential amino acid synthesized from the essential amino acid phenylalanine. Tyrosine transaminase interacts with alpha-ketoglutarate to generate a number of other compounds until homogentisic acid is formed. So at this point, the enzyme homogentisate 1,2-dioxygenase would normally convert homogentisic acid into mallyl acetoacetate. But a genetic mutation on the gene coding for this enzyme renders it inactive and the tyrosine degradation pathway can't proceed. This leads to a systemic buildup of homogentisic acid, or HGA, which is then oxidized via an alternative pathway into benzoquinone acetic acid, or BQA. So the conversion of HGA to BQA is responsible for the symptoms and complications of alkaptonuria. BQA is excreted in the urine, which gives it a dark or black appearance when left standing or when exposed to an alkaline agent. And the conversion of BQA into a melanin-like pigment and its subsequent deposition in connective tissues leads to the bluish-gray deposits noted in our patient, as well as the brown discoloration of her sclera. And deposits in her joints are leading to the arthritis she's experiencing. In alkaptonuria, the amino acid affected by the disorder is tyrosine, which, as discussed above, is considered a non-essential amino acid. The genetic disruption in the tyrosine degradation pathway does not affect the conversion of phenylalanine to tyrosine, which would then convert tyrosine to an essential amino acid. In terms of treatment of alkaptonuria, primary management focuses on symptom management as opposed to interference in the disease pathway and makes the approach much more palliative. However, some studies have shown that the antioxidant vitamin C may reduce the conversion of HGA to BQA, and which is the product responsible for alkaptonuria. The third scenario in this case study is evidence of phenylketonuria, also known as phenylalanine hydroxylase deficiency. So PKU is an inborn metabolic error that where the phenylalanine hydroxylase enzyme is pathogenic and does not code for the enzyme appropriately. Phenylalanine degradation requires PAH and biopterin, which acts as a coenzyme when it's reduced to form BH4. So this coenzyme PAH complex then hydrolyzes phenylalanine into tyrosine. When this hydrolyzation does not occur, phenylalanine is not broken down and will accumulate in the system. Some of the excess phenylalanine will then be deaminated via spontaneous alternate pathways into the phenylketones phenylactate, phenylacetate, and phenylpyruvate, which are then excreted via urine. What is not spontaneously deaminated and excreted will continue to build up, leading to neurologic toxicity, severe cognitive impairment, and epilepsy. It's not quite known how exactly phenylalanine toxicity causes brain damage, but postmortem studies of individuals with PKU show an impairment of neuron myelination, decreased complexity of dendritic branching, and a decrease in synaptic connections. Treatment of PKU focuses on removing phenylalanine from the diet. So as discussed above, as discussed above, we know that phenylalanine is a precursor to tyrosine, which is and is an essential amino acid. But individuals must restrict their intake of phenylalanine, which means that the body can't synthesize tyrosine, turning it into an essential amino acid. And then in terms of a vitamin supplementation, there are no direct vitamin supplements that may decrease instances of PKU or assist in the breakdown of phenylalanine. In the last case study, a woman presents to the clinic with signs and symptoms of megaloblastic anemia. Megaloblastic anemia is a general term that refers to a category of anemias characterized by the presence of megaloblasts, which are abnormally large red blood cell precursors found in the bone marrow. This type of anemia occurs when DNA synthesis when a DNA synthesis abnormality disrupts normal formation of red blood cells. And the primary vitamin deficiency leading to this impaired DNA synthesis is B12 and folate. Folate is required to donate a methyl group to DNA, and B12 is a cofactor that aids in this transfer. Folate is transitioned into a bunch of different precursors, and the conversion. Sorry, let me rephrase that. Folate is required. Folate is transitioned into an intermediate 5-methyl tetrahydrofolate and then back into tetrahydrofolate. When this conversion back to THF is coupled with homocysteine's conversion to methionine, and when B12 is deficient, folate cannot make the conversion from 5-methyl tetrahydrofolate back to THF and becomes trapped. This creates a methionine deficiency as well as an error in DNA synthesis. So this alteration in DNA leads to the miscoding of red blood cells by impairing synthesis of the nucleus of the red blood cell, while the cytoplasm continues to develop normally, which leads to the abnormal size of the red blood cells. Megaloblastic anemia is suspected in this patient based on her presenting symptoms, specifically her neurologic symptoms. So because most anemias target red blood cells and their ability to carry oxygen, they share a series of similar symptoms, all related to increased oxygen support to tissues. So these symptoms include elevated resting heart rate as the body attempts to get more blood and therefore more oxygen to the tissue, fatigue since the supply of systemic oxygen is decreased, and shortness of breath on minimal exertion as the body attempts to take more oxygen in to compensate. This patient is experiencing these symptoms, but also has some neurologic symptoms. Though it's not fully understood why, studies have shown that B12 deficiency can result in neurologic problems, including gait disturbance, dizziness, paresthesia in the extremities, and impaired sensation to vibration. Since B12 is an important cofactor in the conversion of homocysteine to methionine, a decrease in B12 results in a buildup of homocysteine. And a common school of thought is that this buildup of homocysteine leads to neurotoxicity. B12 is also a cofactor in 2 processes that aid in neuronal myelination. Additionally, the patient's GI symptoms also lead to the potential diagnosis of megaloblastic anemia. In some patients, the B12 deficiency seen in megaloblastic anemia results from a pathogenic autoimmune response, where the body forms gastric antibodies that begin targeting intrinsic factor, which is a glycoprotein that binds to B12 and assists in its absorption. And loss of this intrinsic factor leads to a systemic decrease in B12, and one of the resulting symptoms of this process is diarrhea. B12 deficiency also leads to an accelerated metabolism of oral mucosal cells, which leads to the beefy red tongue this patient is presenting with. So all of these symptoms together are highly indicative of a B12 deficiency based anemia, like megaloblastic anemia. But if this patient was suffering from a B6 deficiency, we would see the swollen red tongue, but we'd also see an accompanied mental status change and a pruritic rash. But because she's not exhibiting any of these symptoms, we rule out a B6 deficiency. Lastly, if our patient was suffering from an iron deficiency, which is a type of microcytic anemia, we would expect the fatigue, the shortness of breath, the tachycardia, but we would also expect pallor to be evident in her hands, as well as conjunctiva, and we would expect spoon-shaped nails. Iron deficient anemia results in a smaller mass of circulating red blood cells. And this results in the tissue hypoxia and the pallor most characteristic of iron deficient anemia. In this case study, it also asks the question of how purine and pyrimidine metabolism is affected by B12 deficiency, and the answer is that it's not completely understood, but some studies have shed some light on possible pathways. B12 is a required cofactor in the action of the enzyme methionine synthetase, which causes 5-methyl tetrahydrofolate to accumulate in the cells. 5-methyl tetrahydrofolate is the primary storage form of folates in the cell, and when folate is trapped in its storage form, it leads to a decrease in purine synthesis. Additionally, some studies have aimed to show that B12 deficiencies lead to a reduction in de novo synthesis of precursors of DNA. Finally, in order to appropriately diagnose this individual with megaloblastic anemia, relevant lab tests must indicate confirmatory findings, which includes a complete blood count with a hemoglobin less than 13 in men and less than 12 in women, and a mean corpuscular volume greater than 100. Additionally, a blood smear would show hypersegmented neutrophils, which is a hallmark of a macrocytic anemia. Serum concentration tests should be drawn to determine if the megaloblastic anemia is a result of a B12 and or a folate deficiency. If the results show a B12 B12 less than 200, B12 is the deficient vitamin. If the results show a B12 greater than 400, a folate serum test should be done, and a result less than 4 nanograms per milliliter would indicate a folate deficiency. So the presence of intrinsic factor antibodies could also be indicative of megaloblastic anemia, since lack of intrinsic factor leads to malabsorption of B12.